Thank you, Dr. Wong, for such a generous introduction. It's a great privilege for me to be speaking with you today, and especially following such a great leader in AI, such as Dr. Li Kai-Fu, who is an investor in whose company invested in both Insilico and Deep Longevity, and who I deeply admire and follow. I think that before I start, I wanted to say that I am a deep believer in the power of the China-US relations in healthcare. I think that all of us are fighting the same war on aging and diseases, and that's one of the areas where there needs to be deep collaboration. I'm also the deep believer that the Chinese biopharmaceutical industry is going to develop very, very rapidly, unlike any other industry, and that's going to be great for everybody in the world. So we are very focused and centered around Greater China, and my co-pilot, Dr. Ren Feng, is now setting world records in several areas. So I'm going to talk about some of those today. I'm going to show a lot of slides, so please don't get slide sick. I've got more than 60 slides for only 30 minutes of talk. Some of those claims that I'm going to be making are supported by academic publications. So if you like, please just go to PubMed and search for Insilico plus medicine, and you can see some of our academic work. Or you can, for those of you in the US, you can follow me on Google Scholar. I'm going to be speaking about the end-to-end drug discovery and the value of end-to-end AI-powered drug discovery capabilities. You can also add me on WeChat here. So just a few notes, all the regular disclaimers, not no forward-looking statements, not investment advice in any way. We're doing this to promote science, even though we're a commercial company. Currently, just a couple words about Insilico. So we are about 250 scientists globally. We were founded in 2014, originally at the Emerging Technology Centers in Baltimore. That's the old Johns Hopkins campus. Currently, we're about 250 scientists globally. About half of us are AI/ML. Actually, my background is in computer science, and I switched to biotechnology about 18 years ago from GPU computing, so those semiconductors that are powering deep neural networks today. We also have about 70 drug hunters, and we have over 180 FTEs, biology and chemistry scientists in different CROs. So we act as an operating system on top of many, many labs. And we think about ourselves as a kind of AI pharma semiconductor with many, many layers. So many of you are very familiar with this slide and with this chart. It's pioneering work by Stephen Paul, who pretty much pioneered pharmaceutical analytics, pharmaceutical industry analytics. There are not that many groups that analyze pharma R&D productivity. One is from Tufts University, very famous studies. Another one is Alexander Schumacher. I'm going to talk about his work today. But of course, this was the seminal work by Stephen Paul when he was at Lilly. And he demonstrated that it takes about 12 years for the new molecule from target to hit to submission to launch. And here you can see the probabilities of success at each stage. So you can see that once you know the target, it's possible with high degree of probability to get into phase 1. So most of the failures transpire in the clinical domain and primarily in the phase 2. However, in the original paper, he pretty much avoided focusing on disease hypothesis and target discovery. And that is the area where most of the failures transpire. So biology is very complex. Only about 1 to 5% of the hypotheses and novel target proposals succeed in preclinical experiments. Sometimes it takes a decade to decipher the mechanism of disease, and very often we don't even have the mechanism. Like, for example, Alzheimer's, we still don't know why it happens, regardless of how much money is being spent. Currently, I estimate more than $100 billion that went into Alzheimer's research, and we still don't have a drug. Well, there are some approved products, but we are still unsure about how they work. AI can dramatically accelerate this. Even if we accelerate a little bit, it's still pretty dramatic numbers. So if you look at the traditional pharmaceutical paradigm, and that's what we started looking at when we originated as an AI first company in 2014, started mapping out the different steps that the pharmaceutical company or the entire pharmaceutical process needs to go through in order for the drug to reach the market. And you can see that the target discovery is usually performed by academia, or in some pharmaceutical companies, big pharmaceutical companies, they do have target discovery departments that deal with biology. There are many ways to discover targets. You can throw darts at the, at pathways. You can search Google. You can do literature search. You can do pathway analysis, gene expression analysis, many approaches. But usually that is done by academia and funded by the government. In the pharmaceutical industry, pharmaceutical companies focus on chemistry or biology around those targets to develop new products. And that also involves many steps. And then the products enter clinical phases, and that becomes completely disconnected from the early stage preclinical. So when we looked at the industry and thought about how to enter it, we looked at many areas that can be accelerated by AI. And when Insilico started, we started looking at how to collaborate with many pharmaceutical companies and biotechnology companies in order to accelerate some of those steps with deep learning and deep reinforcement learning, and how to combine them into one comprehensive picture. At that time, most other AI companies were trying to focus just on one single individual step. And for us, we were just providing services to all pharmas. And at that time, they were much more collaborative. They didn't have AI departments. So in many cases, we did really pioneering work. So now I can talk about some of that in the past tense with and talking about successes. So actually, if you are interested in other startups and also pharmaceutical companies that are working in this space and are utilizing AI, there is a really nice academic group in Germany and Switzerland, led by Alexander Schumacher. He publishes usually those features in drug discovery today. You can find a few of them. One very famous one was published last year. It's called Big Techs and Startups in Pharmaceutical R&D. So you can find that paper and look at it. He provides a lot of insights into how other startups are using this, the tools, and also how pharmaceutical companies are using the tools. And surprisingly, you will see that the industry is truly driven by the startups. It's not the pharmaceutical companies that are driving innovation in AI pharma R&D. It's truly the startups, and usually the startups that were born around 2013 to 2015. Because if you were born before 2013 as a company, you don't have a lot of deep learning expertise. You are relying more on more primitive traditional machine learning or computational chemistry approaches, quantum chemistry. At that time, it was very difficult to transform into AI. And companies that were born after 2018, they actually don't have a lot of validation and experimental evidence to provide to the others in order to show that their technology works. Of course, they need to be supported, but nowadays it's much more difficult for them to collaborate with Big Pharma, because Big Pharma has huge departments dedicated to AI. So really the drivers are the companies that were born 2013 to 2015. And there are three types of AI companies. Companies that hunt targets. When we started, there were a lot of companies that were hunting for targets, and now kind of they went away or consolidated. There are many chemistry players and biology engineers designing antibodies, designing small molecule chemistry. About 80% of the companies in our field are focusing just on chemistry, going after more difficult targets. And there are, of course, developers of personalized medicine solutions. So for target hypotheses, there are many ways to identify a target and implicate it in a disease. You can use high-performance robotics, high-throughput screening. You can search through clinical trials data. You can mine PubMed, construct knowledge graphs. So there are hundreds of ways of target identification. And it's very difficult to say which one is better. It's better to look at how all those methods align to provide more evidence for any hypotheses. Usually those companies don't have their own chemistry capabilities and they need to in-license the compounds from other companies. And there are many, many examples where they in-license phase 1, phase 2 compounds and try to conduct their own clinical trials based on the target hypothesis. That's usually the model. There are also many chemistry players. Most companies are taking very well validated targets, a little bit more difficult targets than usual, to demonstrate that they can either accelerate the drug discovery process and do it faster or cheaper, or provide differentiated molecules, or try to drug the undruggables. So far, there have been a few successes there, and you can see that some of the companies even took the molecules into phase 1. We also did that. And our philosophy from the very get-go was to go end-to-end. So we don't differentiate between biology, chemistry, and clinical trials. And when we started, some of the even investors and pharma companies were asking us, okay, so what's your secret sauce? Where do you want to focus? Chemistry, biology, or prediction of clinical trials? So where do we fit you? And we used to say at that time, look, we are the MMA fighter. We really want to go after everything with a defined set of algorithms, deep reinforcement learning, and go end-to-end. And many of them were pretty reluctant to partner back then because they did not believe that it is possible to go end-to-end with one small AI group. But we decided to follow this philosophy from the very beginning and chose only the investors who believed in this philosophy. We also decided to go frictionless. What we noticed is that when you partner with big pharmaceutical companies and you need to do handoffs, so for example, they want to do synthesis or they would want to do biological validation, this handoff results in much friction and also time wasted. And very often, even though their synthetic chemistry might be much better than yours, or biological experimentation capabilities could be much better than yours, once there is this handoff, it creates friction. And usually those partnerships do not succeed or have lower probability of success than when you are in control of the entire process with many CROs. And also we believe in highly distributed, highly parallel validation experiments, meaning that we would do the same experiment in several labs in parallel, and we will not wait up until one experiment completes to start the other one. That means that many of those parallel experiments, sometimes you see which lab is better, which lab is worse, and you always have redundant data. If you fail somewhere, you learn as an AI company. If you succeed, you have a much stronger data room than if you were to perform it in just one lab. And it's one of the ways to also test a lot of CROs and test a lot of preclinical models. So one of our first experiments that we did, we can actually refer to this publication. Again, we try not to make any claims until it's backed up with a published paper in a peer-reviewed journal. So 2015, we partnered with a company called Biotime, and they challenged us to find new targets in the embryonic stem cell data, embryonic cell data that was for embryonic cells that were differentiated into multiple cell types, mesoderm, ectoderm, endoderm, into many, many different cell types. They took gene expression data from every step of differentiation. They had a few thousand data sets. So we trained deep neural networks on that data to predict the differentiation state and identified a really cool feature called SCOX7A1, a protein that we implicated in embryonic to fetal transition, performed the experiments and published. But since it took 2 years of experimental work, we didn't publish it in Nature or some other like top journal. We published it in lower tier journal. Based on this work and also, of course, on other IP that Biotime created, they generated another company called Ajax and then listed it on NYSE. So we realized that there is value to be created from target ID, and we got very, very little payment from that. So we decided to also look at how we can create shareholder value by utilizing our target ID capabilities. And to do that, we decided to develop our own chemistry capabilities, because if you own your own chemistry, you don't need to in-license. You can go end-to-end and be in control of those molecules. Because when you in-license, it's kind of it's a liability. So you need to progress the molecule, and if you fail somewhere, or if there are any delays, the company that is licensing the drug to you, they might take it back, right? Or you see that it might not be as profitable anymore, and you want to give it up. It's a liability. So, but if you own your own chemistry, you can tailor the molecules with the desired properties, and you can own your own chemistry, and you can do whatever you want with it. So we decided to go chemistry, and our first papers in chemistry came up in 2016. So we submitted our first paper called Cornucopia of Meaningful Leads in 2016 into a peer-reviewed journal. Again, not the top, but we wanted fast. It was picked up by some of the really top AI scientists at that time. Here are some of the top two fathers of AI. This paper came out in November. It got popularized in January 2017 and became pretty famous. And this is the chart that I'm very proud of. So this is the history of deep learning and deep reinforcement learning of basically AI. The true revolution started around 2013, 2014. And in 2014, GANs were published. That's generative adversarial networks, kind of a form of AI imagination. And we picked up on this technology and decided to use it for chemistry. We struggled for a couple years, but then published our first peer-reviewed paper in 2016. So submitted it in 2016 in June. And then another group led by Alana Spurogudzik out of Harvard put on archive a paper on variational autoencoder. So ours was in review, so we were very sad at that time because we were still waiting for the paper to be reviewed and published. And then it got published. So this one is a little bit more popular, and we used to be kind of competitors with Alana Spurogudzik. Also, we published a number of other theoretical works, and in 2017, 2018, we published our first experimental validation of JAK3 selective inhibitors in one of the ACS journals. But it was a pretty quiet paper, even though we did the validation at WuXi. And then WuXi AppTech, a wonderful company that I deeply admire, decided to do a race with us and nominated a target called DDR1, very well-known target. So we could go and apply our AI to very rapidly generate new hypotheses and new molecules with the desired properties. Again, lots of information was available about DDR1 kinase. Very quickly, we generated 30,000 structures, narrowed it down to 40, synthesized 6. So the entire experiment took 46 days, and we actually collaborated with Alana Spurogudzik, who used to be our competitor before, right? So I actually just spent a few days in his lab last week. So he is our scientific advisor now, and we think that his group is top 1, top 2, depending on how you compare with us. So it's great to collaborate with him. And in that paper, we demonstrated that we can synthesize those 6. 4 worked. Of course, out of those 30, many were completely novel. It was very difficult to synthesize, so we had to prioritize for synthesis. Took just 2 of them into microsomal assays. Took 1 of them into mice. Again, think about it. We wanted to do we wanted to show acceleration, so we had to prioritize for synthesis. So whoever kind of looks at this paper and criticizes, do it yourself. Let's see it, because you have to prioritize synthesis as well. But that was a pretty famous paper. It went viral. So here is the CEO of Novartis, Vastar Vasimkhan, presenting the paper in 2019. And person who sent me this picture is Michael Antonov, who is the founder of Oculus, those metaverse beginning. And he was at this lecture, and he sent me this picture. But then we managed to integrate many of those models, not only one, like Gentrol, into a system called Chemistry 42. This slide shows the evolution of this system. So this was the model that we published. But later we integrated more than 30 models into Chemistry 42. We also provided very substantial virtual screening capabilities in there. And many of those are experimentally validated. It's usually costs you about like $250,000 to even $500,000 to validate just one properly. So it's very expensive to build those systems because then medicinal chemists are using them. Now our software is deployed in 9 out of the top 30 pharmaceutical companies, so it's pretty cool. And we opened it for big pharma around 2021. Now we have 3 pillars of our pharma AI platform. One is called Panda Omics. It identifies new targets very quickly and integrates more than 60 target discovery philosophies. If we have the time, I can show you how it works. So to prioritize the targets that already exist or find the new ones. Once you do that, we can very rapidly use chemistry 42 to design new molecules with the desired properties. And currently we are working on what I call the grand challenge of drug discovery and drug development. We're working on a predictor of clinical trials outcomes from phase 2 to phase 3 based primarily on preclinical data. So we look at how implicated the target is in a disease, how heterogeneous the target is in a patient population, and then the clinical study design. So this system is 5 years in the works already, and we still haven't released it. We hope to release it this year. We want the others to use our tools. And you know that target discovery is more of an art than science. Usually big pharmaceutical companies, they claim to go after novel targets. They really want novelty. But at the end, if you show them a novel target, they'll say, well, see you at phase 2, right? Because they want to de-risk the programs. And that's why they usually go for more conservative targets, also from the AI companies. What we are trying to do with our AI is to increase the confidence by increasing the novelty at the same time. And the way you do it, we integrate multiple data types for target discovery. So omics, we have about 10 million samples there. With different data types. So novelty comes from omics, and then you use text and even financial scores and QOL scores to increase confidence. We even score scientists by their credibility to understand how credible the research is and which one to prioritize for target ID. Then you use many AI modules. It's a diverse set of AI algorithms to identify novel, safe, and druggable targets. You give it to chemistry 42. I'm not going to cover this module, but basically think of it as a black box that can generate small molecules with the desired properties very quickly. Internally, we integrate all of those 3 modules so they work seamlessly. But you know, since in pharma those areas are disconnected, you need to offer those 3 areas separately. And recently what we did, we showed how those 3 modules can work together in unison. Actually, if you want to learn a little bit more about those experiments, you can, and if you're in the US, you can use YouTube and watch a lecture by Michael Levitt, who got his Nobel Prize primarily for folding on computational biology. So he talks about this work. He's our scientific advisor since 2014, and he is the co-author on this paper. We just put a paper on preprint server, which shows you how we can use AlphaFold crystal prediction tool for drug discovery. And what we did in that experiment, we took hepatocarcinoma, so liver cancer, very rapidly identified some targets that are present in AlphaFold database, but are not do not have experimental structures. Zoomed in onto CDK20 for many reasons, gave it to chemistry 42, did new molecular synthesis, did bioassay testing. So it kind of shows you the value of AlphaFold. There are no shortage of great targets with good structures, but here we just wanted to show how to apply AlphaFold for this purpose. With the CDK20 AlphaFold structure, the structure-based drug design using chemistry 42, synthesized and tested in 30 days, very rapidly, under the leadership of Dr. Ren. He is the first author of this paper. By the way, it's 100% done in China. So the entire end-to-end. And even Michael Levitt went there for a few months and worked on this paper. So we very rapidly managed to go for weak hits, 8 micromolar. But those are not exciting to medicinal chemists, right? And then we took another 20 days and used one of the hits to optimize and got to 200 to 300 nanomolar hits. So those are very respectable, novel, and pretty selective. So we're still working on experimental evidence to be able to submit to a high journal. But currently, we just updated this archive preprint. And in 2020, we decided to, so this is a pretty cool success story that I'm deeply proud of. So in 2020, we decided to go big on and zoom on a grand challenge. So novel target, novel molecule for a broad disease with no cure. Can we combine biology and chemistry together and go after a big disease? So to do that, we decided to also become a different company. So we decided to be an AI-powered biotech and not AI-first company anymore. So we had to build the drug discovery team that sits on top of many CROs. So it became an yin-yang of AI and drug discovery. And to do that, we partnered with Dr. Ren Fang. So now, I was very afraid somebody with big pharmaceutical background and with many, many years at CRO. So he also was working for Medicinal for many years. It was part of the big part of their success. And, PhD from Harvard. I thought that, okay, can he truly embrace AI and accelerate? And in record time, he built an amazing team of drug hunters, so head of biology, head of chemistry in Shanghai, and embraced AI and turned it into the tool that we can all use to accelerate. So his nickname at the company is the Magician. So he really turned our AI into magic. And we zoomed into IPF, so fibrosis. We found some targets that were implicated in fibrosis and aging, decided to purpose towards IPF. Big market, $3 billion, 2 approved drugs. One is called nintedanib from BI, one is called Esbriet from Roche. Very rapidly, we identified targets that are implicated in aging and fibrosis using deep neural networks. The basic bioinformatics. I use text databases. I have one of the largest grants databases and text databases on the planet to assess novelty. Prioritized 20 targets, tested 5, all 5 worked. Zoomed into 1, generated molecules, conducted multiple assays, went into mice, implicated the target in lung fibrosis, liver fibrosis, skin fibrosis, kidney fibrosis. And skin fibrosis were already at mice as well. Very rapidly generated 79 compounds. So you can see the distribution. Most of them were 10 to 100 nanomolar. Many of them were under 10 nanomolar. And this is 055. This is our this was our lead. Now it's our then it became candidate, and now it's a drug in clinical trials. And we also have 073 for kidney fibrosis. Performed multiple experiments. So here is a lung function experiment to show comparable activity of our compound and different mechanisms of action at 1/10 of the dose of the current best in class market standard. Also conducted multiple other experiments. So here we used IPF donor cells, demonstrated fivefold more activity in fibroblast to myofibroblast transition than nintedanib, and 16-fold more potency than nintedanib in epithelial to mesenchymal transition. So those are very common assays. And then we nominated preclinical candidate last February 24. Many experiments completed. And to my knowledge, that was the record for combining novel target discovery with chemistry and claiming PCC. And then we also did the additional experiments to do the IND filing. So did a lot of talks. Also, in the same time, nominated preclinical candidate for the same target, actually one other target for kidney fibrosis. And last November 30th, last year, we went first in humans, started phase 0 microdosing trial, because it's a new target. You need to be damn sure that you calibrate the dose, basic safety, understand the distribution. And using the same kind of paradigm, we created a pipeline of drugs. Now we have over 30 programs internally, different stages, and some are progressing very rapidly. So now we have one in phase, well, back then we were in phase 0, and nominated multiple preclinical candidates for also anemia, IBD, immuno-oncology. So this one, we partnered with Fosun Pharma, an amazing company in China and globally, and progressed it to PCC as well, just announced. And 2 days ago, we've announced that we've completed the phase 0 microdosing, no surprises, and entered phase 1 with a fully AI discovered drug. To my knowledge, that's the first. And also progressed into phase 1 and progressed into IND enabling with Fosun on QPCTL asset. So now we want to make sure that our software is available to the others. And we are offering our PandaOmics and Chemistry 42 systems for out-licensing. I don't think I have enough time to show you how it works. I have a screensaver. You can find it online. So if you're interested, and you can contact me, I can send you the link to the screencast. So here I would like to open the floor for questions. I know that I'm out of time.